Good morning. I wanted to welcome you to our Spotted Wing Drosophila webinar, Good Bugs and Bad, Using Biological Controls for Spotted Wing Drosophila Management. This webinar is a presentation of our Sustainable Spotted Wing Drosophila Management Specialty Crops Research Initiative project. This project is funded through a USDA NIFA Specialty Crops Research Initiative grant, and we'll have a little bit more information on that at the end of this seminar. I'm Hannah Burak, and I'm the project director of this grant. My role in today's session is to welcome you, give you a brief overview of our larger project, and then introduce you to the rest of our panelists for the remainder of the day. A few notes on mechanics before we get rolling. If you have questions during the webinar, please type your questions into the chat box, which a few of you have already been using, and our moderator will collate and ask these questions either during section breaks or at the end of the session. As a participant, you are muted, so you can't ask your questions out loud, so please type those in the chat box. We are also recording this webinar to post online at our project website and on YouTube after this session. So if you're watching a recording of this webinar and you have questions as you're going, those can be emailed to me, Hannah Burak, at NC State University at the email address on the screen. Our presenters for today, myself, Hannah Burak, I'm at North Carolina State University, and again, I will be giving you a brief overview at the beginning of this session. Our other presenters are Kent Dana, who's a faculty member at the University of California at Berkeley, Kim Holmer, who's a research scientist at the USDA ARS based in Delaware, Heather Leach, who is a PhD student at Michigan State University, and then our moderator is Lauren Diepenbrock, who is the project director of the Specialty Crops Research Initiative funded grant here at North Carolina State. Their photos are here on this slide, so you know what folks look like. And as we move through the presentation, you'll see the photo of the speaker up in the right-hand corner of your screen. So that'll give you a sense of who's talking to you throughout the course of this presentation. As I mentioned, this webinar is part of a larger National Spotted Wing Drosophila Research and Extension project. I'm the project director based here at North Carolina State University, and listed on this slide are the project directors based in other locations, our co-principal investigators, our project coordinator, and then our affiliations down at the bottom of the slide. The reason why I wanted to share this information with you is to both illustrate the scope of this project and to highlight who in your region or state may be working on spotted wing Drosophila as part of this collaboration. I encourage you, if you're based at any of these locations and you have additional questions, to please reach out to our collaborators at these states. The overall goal of our larger national project is to integrate spotted wing Drosophila management practices with those necessary for other pest species. As those of you who are joining us understand, spotted wing Drosophila has been a game changer in berry and tree fruit production. And it's necessitated an urgent and large response, but at the same time, it has impacted management of other pest species. We also want to reduce the reliance on insecticides as our only means of spotted wing Drosophila control. In the short term, that has been our immediate response. But in the long term, we want to have an integrated approach that implements cultural, biological, and chemical control tactics. Next, we are delivering this information to our stakeholders, our growers, our extension personnel, our fellow researchers and extension scientists, and to facilitate adoption of the recommendations that we develop. So that's what we're doing today. We're sharing with you some of the results of this project, and you can read more about the results of this project at our website, which is highlighted there on the bottom of the screen. There are three large overall objectives of this project. The first is to implement and evaluate spotted wing Drosophila management programs. The second, to develop tactics and tools to predict spotted wing Drosophila risks. And the third is to optimize spotted wing Drosophila management programs. There's a number of sub-objectives under each of these large objectives. And today we're going to be focusing on sub-objective associated with objective 3, which is to discover natural enemies capable of reducing spotted wing Drosophila populations. That is our topic for today's webinar. Here's our outline. You've heard from me. I'm going to be stepping back for the rest of the session in just a few minutes here. I'll be turning it over to Heather, who's going to talk about the current status of biological control for SWD in the United States. Then Kent will talk about efforts to implement classical biological control. Kim will talk about the importance of identifying what natural enemies we are dealing with. And then finally, Kent will wrap up talking about what's next, how we use the information that we've been generating to help improve spotted wing Drosophila management. So with that, I'm going to turn off my video so you're no longer going to see me talking. And then I'm going to turn it over to Heather for our first session.
Great. So thanks, Hannah, for that introduction. And I should be able to proceed through the screen here. So as Hannah said, I'm going to be talking about endemic biological control. So that means what's already here in the US that might be helping us control spotted wing. So I'm going to first be talking about predators and understanding what sort of predators might be attacking spotted wing Drosophila out there already for us. And before I talk about that, I want to talk about the life cycle of spotted wing with this. And Hannah, I might ask you to take over the slides here since they're not really. Thank you. So again, I want to talk about the life cycle of spotted wing Drosophila in the perspective of a natural enemy. So we know that these adults are out there laying eggs in the fruit, but the adults are fairly mobile. So we really want to focus on those larval stages and those pupal stages and understanding how we can best target those. And when we talk about the pupae, the next slide. We know that roughly 80% of the pupae are found within the soil. So most of them, those late larval instars, will drop out of the fruit and then pupate on the ground. And the majority of those pupae are slightly buried. So in that photo there, you can sort of see that orange outline. That's that pupae that's slightly buried in soil there. So again, regardless of fruit type, we know that the majority of those pupae are within the soil. And so what we're finding is that those pupae are considered what we're thinking of as vulnerable. So these adults are mobile, but and those larvae are kind of cryptic, right? They're hidden in that fruit. But again, those pupae are outside of the fruit and considered vulnerable. And we also know from modeling that about 60% of the population of spotted wing Drosophila is larvae and pupae for the majority of the year. So again, that's really what we're keying in on and want to target when we talk about biological control. So again, as I said, I'm going to focus on predators, and the states you see highlighted here are those states that have done some research on predators on spotted wing Drosophila. So I'm going to talk about those as we go through here. So what I also want to point out, this is some research that was done in Maine, and we're finding that the majority of our predators are indeed arthropods, so insects and spiders, and we're finding that there's greater predation when those pupae are left on the surface compared to if they're buried slightly below the surface. So on this graph here, you can see the percent predation of SWD pupae, and that black bar is when they're left on the surface. So we have two different sites that are highlighted here. So you can see at site A, we do see a big drop off when those pupae are buried. At that second site, site B, in both years, we see that those predators seem to be a little bit hungrier. They're willing to dig below the surface to get those pupae. But again, we do know that the best chance of predation is when those pupae are left on the surface. And what we're finding is that the most common predators are these crickets, daddy longlegs or harvestmen, ants, and ground beetles are really abundant in these areas. And when we leave pupae out and find out how many predators are actually active in those areas and taking them away, we're finding that up to 90% of those pupae can be removed. And these were done in blackberry and blueberry crops. And so we're actually getting pretty good predation when we're controlling for this and watching for this. And we're also finding from some really cool research when we actually videotape those pupae that are left out, ants are actually coming in and digging up those pupae and then carrying them away. And so that happened in this one study on 39 occasions. So we know that these predators are learning to adjust and learning how to get this resource of spotted wing Drosophila pupae. And we also have done some research on understanding what sort of habitat might best support these predators. And so we did this mulching study in blueberries in Michigan. And what we were finding, depending on different mulching, so we had a buckwheat, a wood chips, a bare ground, or a weed fabric treatment in these blueberries. And we found that regardless of the mulching type, we found similar predation of the pupae in all habitat types. So you can see below each of those pictures, we have a little petri dish. And in each one of those petri dishes, we put out pupae to see how many were removed by those predators. And so again, we saw no difference between these treatments. However, if you move to the next slide, you can see that there's some predators that prefer certain mulches. So there's a lot of stuff going on in this graph here, and really what I want you to focus on is the fact that when we look at all predators across all of these different habitat types, we don't see much of a difference, so in that first group there. But as we go across some of these different predators, we're starting to see differences in what these predators might prefer. So for instance, ants seem to prefer that bare ground treatment versus ground beetles seem to prefer that buckwheat and weed fabric treatment. So really this is mostly a lesson of knowing what you have on your farm and what you might be supporting on your farm with your different mulch types. I also want to point out that we're finding all over the place is that these farms that are managed in a conventional way have reduced abundance of natural enemies. And this is true with predators in blueberry, for instance, and this work was done out of Georgia. So what we're finding is that unmanaged sites have the most predators, and then those organic sites have some intermediate level of natural enemies on the ground. So this isn't surprising to us, but this is something that we should certainly be applying to spotted wing Drosophila management when we think about what sort of environment they're in on our farm and whether or not there's likely to be predators in abundance there. So overall, with predators, what we're finding is that those SWD pupae outside of the fruit just below the soil surface. So again, we know that that's considered somewhat of a vulnerable state for them. We also know that those predators can consume greater than 60% of the pupae on average. So we're finding good predation when we look for it. However, we also know that fewer predators are found in those high input conventional farms. So if they're offering control for us is probably unlikely because we are spraying a lot for control of spotted wing Drosophila. So next I want to move on to talking about parasitoids. So what naturally occurring parasitoids are there already in the United States that might be helping us control this pest? So again, these states highlighted are the states that we looked at parasitoids that are already in this region. And before I go into these, I kind of want to get you used to these scientific names since these are a little bit complicated. So we've found 5 naturally occurring parasitoids in the United States. And so there's 3 larval parasitoids that are really abundant and 2 pupal parasitoids that are really abundant. So those larval parasitoids will be indicated by that L after their name, and then the pupal parasitoids will be indicated by that P after their name. And I don't want you to get too overwhelmed by these scientific names here. So we have this Leptopollina, the 2 Leptopollina species, an Asobara species. You don't need to pay too much attention to that. And then we also have these 2 pupal species. Those I kind of want you to pay a little bit more attention to. So you'll hear me say these a lot, Pachyropoideus and Trichopria. And then I also want to share a quick video of you just so everyone can get on the same page of what these parasitoids are doing. So one parasitoid is laying 1 egg in a pupae or a larvae. So here you can see this is one of our pupal parasitoids, and she's depositing an egg into an SWD pupae. So this can also give you a good size reference here. So these parasitoids are very small, and they're attacking these spotted wing Drosophila, either larvae or pupae. And when we talk about SWD parasitoids, we can't talk about them without talking about this idea of encapsulation. And what that basically means is resistance to parasitism. Spotted wing Drosophila have a high resistance to parasitism. And what that means is when they're attacked, especially by those parasitoids that attack the larvae, they have this ability to basically smother that parasitoid egg to death. And what you'll see is that blackened area on the larvae, and you can also see that through the adult where the arrows are pointing there. So compared to other Drosophilids, we know that SWD is highly resistant to parasitism, and that'll be an important theme throughout the rest of this year. So I want to talk about what we're seeing in each of these states, just a quick overview of what parasitoids are abundant. So if we focus in on North Carolina, we're seeing that the majority of the parasitoids we find are these Leptopolina species, and that's a larval parasitoid. And then we're also getting those two pupal parasitoids, those Pachyropoides and Trichopria. And in this same North Carolina study, they found that dependent on location is a different number of parasitoids, which we would expect. So what we're finding in this graph here, you can see the number of parasitoids. There's more parasitoids found in non-crop habitats than there are in the crop. And I also want to point out, if you notice this green bar, that was in the crop at one site. Where we're getting about 45 parasitoids in that survey. However, when we were looking at that site, we only found about 2.5% parasitism. So even though we're getting an abundance of parasitoids, our levels of parasitism are quite low. In Virginia, we're finding a similar community. So this graph here is showing the number of parasitoids we see, and this is distance from crop edge. So that 1 would be closer to the crop edge, and that 4 would be in the crop interior. So again, what we're finding in that blue is mostly that Leptopolina species, or that larval parasitoid. And then we're also finding a few of those Pachyropoides species, or that pupal parasitoid. So overall, what we're finding here is that there's more parasitoids closer to the crop edge, and those larval parasitoids seem to be more common than those pupal parasitoids. In New York, we're finding a pretty similar community, except we're finding a different larval parasitoid. So those blue bars indicate a larval parasitoid called Asobara, and then again, we're finding a pupal parasitoid, Pachyropoides. In Michigan, we have a really similar community as we do in New York. So again, we're finding that Asobara species, especially in August and September. And then later into the season in October, we're finding that Pachyropoides species. So if you'll notice, this Pachyropoides species is coming up over and over again. We're finding this one very commonly. And if we jump over to California, what we're finding, they did this a little bit differently, but what they were finding in raspberries here is in the field and in the riparian area of raspberries, they were finding that their percent parasitism was around 12%. So they're actually getting pretty good parasitism. And when they were looking at what parasitoids were actually causing that, they were finding that the majority of that is caused from that Trichopria species, so that pupal parasitoid, which you can see pictured in that top picture there. They were finding a little bit of parasitism from that Pachyropoides species and those larval parasitoids, but again, the majority of the parasitism there is from that Trichopria species. So if we look across all of the United States and in these states that we've done these surveys in, we found those 3 common larval parasitoids and 2 common pupal parasitoids. Those larval parasitoids, while we're seeing that they're abundant, we don't see that they're really attacking spotted-winged Drosophila in a great number. So our best candidate endemic parasitoids, or naturally occurring parasitoids, are that Pachyropoides species and that Trichopria species. So Pachyropoides is considered a cosmopolitan species. It has a greater known distribution and this ability to withstand lower temperatures and higher temperatures. So we're more likely to see it in these states that we're surveying for. That Trichopria species we haven't seen as often, however, we do know that it's contributing to a greater amount of parasitism on SWD pupae. So for the rest of this talk, my section of the talk, I'm just going to focus on those 2 pupal parasitoids, that Trichopria and that Pachyropoides species. So here's kind of a complicated graph, and I'm just going to walk you through this here. So here we can see Drosophila suzukii, or spotted-winged Drosophila, is highlighted in red on the left. And all these other scientific names going up or down are species that are closely related to spotted-winged Drosophila. As we go across, we can see that the number of offspring produced by these different pupal parasitoids in spotted-winged Drosophila is pretty good. So we're getting parasitism from SWD focused on that red bar. However, when we look across to all those other closely related species, we see that Trichopria and Pachyropoides are both attacking all other related species. So they're not host-specific, and they're not just keying in on spotted-winged Drosophila. We also know something about their efficiency. So in lab studies, we've seen that Pachyropoides in the red there, or Trichopria in the blue, has good parasitism whether the pupae are located in the fruit or in the soil. We do see that Trichopria is a little bit more efficient. So with Pachyropoides, we see that there's a drop-off of pupae in the soil compared to pupae in the fruit. So they seem to struggle a little bit finding that pupae in the soil. And we also know that these parasitoids are capable of laying 6 to 7 eggs per day. And as these females age, these adult female parasitoids age, they decrease in their egg production. So overall, we know that this Trichoprea species is more efficient than Pachyropoides. We also have to wonder something about what sort of resources these parasitoids might need, and this is drawing back to what you might need to do on your farm to help support these natural enemies and then other natural enemies we'll talk about later in this webinar. So we know that these parasitoids need some sort of sugar resource. So this study here is their longevity, if they were either fasted or just fed water, or if they were fed honey and water. So we know that if they're fed honey plus water, their longevity increases by 50 days. They're able to lay or live for around 60 days. And in this lifetime, they can parasitize around 300 to 450 pupae. And we also know, looking at these lab studies, is when we're giving them honey and water, we can see that their ability to parasitize these pupae increases by greater than 25%. So again, thinking about what sort of resources they might need on your farm is an important theme here. So overall, we're finding that Trichoprea is more effective than Pachyropoides. However, it's less tolerant to both these high and low temperatures. So again, we're less likely to see this Trichoprea spread throughout the United States. I also want to talk about, again, what sort of things these parasitoids might need. And this is really talking about habitat and refugia needed on your farm. So from some studies we've done in the lab, we know that Pachyropoides is really sensitive to insecticides. So this was a lab study that was done out in Oregon. And again, so we treated these parasitoids with either Mustang Max or Malathion. And we saw that they had up to 100% mortality for 25 days after an application of these insecticides. So again, when we're thinking about the way that we mostly manage spotted-winged Drosophila, which is with these chemical applications, they're unlikely to really thrive in your crop. So we need to pay attention to what's surrounding the crop and what areas might be safe for these natural enemies. We also find through our research and our surveys that more parasitoids are found in those riparian or wood edge habitats, and more predators are found in those organic and unmanaged farms. So this is a common theme as we go through here. And also I want to point out that from some modeling that was done on spotted-winged Drosophila populations, here you can see these lines indicate the overall population of spotted-winged Drosophila as we gain cumulative degree days. And this 2% parasitism is that dashed line there. So percent control of the population with 2% parasitism is pretty low. It's not really contributing much to the population effect. However, when you increase to 15% parasitism, that's where you're seeing that greater effect of the population. So really what this means is that our current biological control is unsuitable. So supporting natural enemies on your farm might help on a local scale. So thinking about what sort of resources they might need and how you can support those might benefit you, but especially later in the season, that population level effects are unlikely to be affected by what's these naturally occurring natural enemies. So really this justifies our need for foreign exploration and classical biological control. And with that, I'll turn it over to Kent, who will talk about just that.
Thank you, Heather.
All right, we're going to pause quick while I give you control, Kent, and let you advance to the next slide. And we did have one question that we wanted to ask from the chat box.
All right, Kent, actually, can you? We had a question that All right, can you hear me? All right.
Either Hannah or Lauren can speak.
Hannah, you're gonna. The question is, you're going to talk. It's not letting me.
Question is, how do either entrust or pyganic sprays impact the naturally occurring parasitoid numbers? And Heather, could you answer that for us briefly?
Sure. So we haven't done any thorough look at how these parasitoids are affected directly by these sprays. So again, we did that Mustang Max and Malathion application. This is something we're hoping to look at more in the future here. But again, from our field studies, we also know that we're just finding more parasitoids in these organic farms and in these unmanaged farms. So using Pyganic and Entrust is likely to have less of an effect on them than these what we consider harsher chemistries.
All right, so Kent, would you like to take us to the next slide then?
Let's see if it works. It's not going down yet, Hannah. Please advance, thank you. So I'm working with a lot of people on classic biocontrol. And I want to start by pointing out that this is really a large global project. Next. I'm based in Berkeley. We're doing the quarantine work from here. We're also working closely with a group from Oregon State led by Von Walton, a group at USDA, Kim Homer, Matt Buffington, a group in Italy, and they've been collecting with us in China and South Korea. There's also another group out of Switzerland working CABI, Tim Hayes in charge of that group. And we're really all focused on collecting material in China and South Korea. Next. Heather did a really nice job of summarizing the different natural enemies that attack spotted wing in North America. What's important to point out is that even though we've got these larval parasitoids that are being found in your fields, a lot of them are being reared from other fruit flies, such as melanogaster, not spotted wing because of the encapsulation that Heather talked about. This is the resistance that these natural enemies find when they attack spotted wing Drosophila. So our goal is to find other larval parasitoids that might work to help us control spotted wing. Next. So this pest is considered to be native to Asia. From the literature, we know that there are parasitoids in Japan. We believe it might be from China. And we also know from literature that there's some natural enemies in South Korea. So these are maps of South Korea. We can see the different colored dots are the different collection sites we've gone to. In South Korea, we've collected in 2013, 2014, 16, and 2017. In China, we really focused on southern China, and we had a collection in 2017 where we brought back material. Next. This work could not be done without cooperators in this area. So if you go around clockwise, we see a group of students from Seoul University that worked with Peter Scheer and Jeff Miller on one of our first collection trips. Below that, there's myself with Johan Son in South Korea, Helmut Riedel, Betsy Miller, and Jeffrey Miller in South Korea. And up above, this is our collection in China this last year. We've got collaborators from China, Xingqing Wang, who is a research specialist in my lab, Massimo from Italy, and myself. And this is one of our more effective collections that we made. Next. Where are we collecting? We're not really collecting in growers' farms. We're actually going to hillsides. For the most part, we're going up to higher elevations. And we're looking for natural vegetation, resident vegetation. So for example, this is Massimo and Xingqing collecting from wild berries in China on the hillsides. So we're usually around 2,000 to 4,000 feet up in elevation. And it's not unusual for spotted wing to start up at higher elevations and then to go down into the valley where the crop is found. In China, I want to point out that they do consider spotted wing to be a pest in some of these regions, where in South Korea, they don't consider it to be a pest. Next. This is just showing a close-up of some of this wild rubus where we're collecting. We also have got other host plants, both in China and South Korea. Next. This is what we just call Western strawberry. I'm going to pull back because remember that previous slide showed a large raspberry bush. Next, these Western strawberries are actually growing like a clover on the ground. We see Xingqing and Wei Yi here collecting. This is important for us to consider because these parasitoids are both on the ground and up in the tree, in the raspberries. Next slide. We can just see the size of these western strawberries, very, very small, being held in the hand here. Next. And we're also finding in these large bushes called sambucus. This is from China. So this is important for me to think about because these parasitoids don't have an affinity just to one particular host plant, and they're found in low-lying plants as well as plants that are growing relatively high. Next. We take this material we're collecting. We've collected over 100,000 fruit from these different years and different sites. Sometimes we're lucky. This is Kim Homer and Emilio in a laboratory in China, working with our cooperators there. So in this situation, we had a nice setup. Other times, next slide, we work out of hotel rooms. This is in South Korea. And when you get in a situation like this, you just ask the maid not to come in because you're using the bathtub to hold rotten strawberries as you're going through samples. Next. So the important thing is, what are we finding in these different sites? We're finding all kinds of different parasitoids in South Korea and China that we're not finding in North America or Europe. The range has been quite impressive, but I've got in bold here 3 different species. The first is a braconid, Asobara japonica. This is one of the most common parasitoids we're finding. The second 2 are fecundity. As Heather mentioned, there are no common names. So this is Ganaspis brasiliensis and Leptopolyma japonica. I'm going to spend most of the time talking about these 3 species. And when I come back and talk about quarantine work, they will be the stars of what we hope to be a foreign exploration and release. We're also finding the pupal parasitoids, Pachycropoideus and Trichopria, but we're not as interested in importing those because they're already in North America. So these are close-ups of these 3 species, the 2 fecundity, Ganaspis and Leptopolyma, and the braconid, Asobara. We're finding these both in China and South Korea. Asobara comes in a little bit later. We're finding it more commonly in rotted fruit. The 2 fecundity, the 2 up in the upper part of the graph, those we think are probably going to be more important. We're finding those early in the season. We're finding these in fruit that's just ripening, and they seem to be more specialized, which I'll talk about in the next session. So one of the things that we wanted to do is to compare these annual collections. 2013, 2014, we're putting out fruit in traps and looking to see what natural enemies are attacking the fruit flies that come to that. So for example, we put out banana sections. We're getting a lot of Asobara, some Leptopolyma, a lot of the pupal parasitoids in these 2 early collections. In 2016 and 2017, we're collecting fruit from collection sites. We're going in earlier in the season, and we get a shift of parasitoids to these 2 fecundity, especially Ganaspis. And that's one reason why we think that these are probably more specialized on spotted-winged Drosophila than these other braconids, such as Asobara. Well, important question. What levels of parasitism do we expect we'll get in the United States if we're able to release these parasitoids? From 12 sites in South Korea from a 2014 collection, this is when we're collecting in about late June to early July. We're getting both braconid, that's the Asobara, and the fecundity, Leptopolyma and Ganaspis, and we're getting up to about 20% parasitism. In China, our collections were much, much better. So here we're getting anywhere between 3 and 73% parasitism at these different collection sites. Are we going to expect 73% parasitism throughout the United States? Probably not, but we're going to see a range depending on how these parasitoids adapt to each region. But as Von Walton's model showed, even a small reduction of 10, 15, 20% might really have a large impact on the overall population. So what we need to do now is to bring these things back into quarantine and to look to see their biology and especially their taxonomy. And that's where Kim Homer is going to take this over.
All right, Kim, I'm going to give you control if you want to try to advance. Otherwise, I can advance for you. All right, Kim.
There we go. Okay, now I'm unmuted. I don't see the advance bar, so I'm going to ask you to advance for me.
Will do.
I don't think this. I'm at the beginning of my series here. The first slide should be the role of taxonomy in successful classical biocontrol. It looks like some of the slides were dropped maybe, or maybe moved out of order. Well, we'll go ahead with what we have, and I'll see if I can adapt the order. I'm going to talk for a few minutes on the importance of taxonomy in helping us identify the species that we're finding through foreign exploration and finding as native species in the U.S. And as many of you probably know, taxonomy is the branch of biology that deals with the description, classification, identification, and naming of species. And this is very important for us when we have a diverse group of natural enemies. We have to know what we're dealing with and how to classify them properly. We know that there is actually quite a bit of research on Drosophila parasitoids in general. Many of you probably know that Drosophila is somewhat of a lab rat in biology, and there have been many, many studies in ecology, behavior, genetics, and so on that have been done with Drosophila species. And some of this work, some of these species also have been used to conduct a lot of basic studies with parasitoids of these Drosophila species. Next. But in fact, very little of this research over the past years was conducted specifically with natural enemies of spotted-winged Drosophila. So we've really begun working with spotted-winged Drosophila natural enemies for this particular project, as spotted wing has established in North America and then also eventually in Europe and other countries. As Kent has mentioned already, foreign exploration has shown that there's quite a diversity of natural enemies attacking spotted wing in China, particularly parasitoids. And now, because we've been working with taxonomists who specialize in these groups of natural enemies, we believe we have a pretty good understanding of what some of these species are. Next. Two of the species that Kent mentioned that we're particularly interested in are this Canaspis brasiliensis and Leptopolina japonica. And you can see that these are very, very tiny insects. Their size is shown by the millimeter bar on the dime. Even trained entomologists have difficulty identifying species like this based simply on their appearance. So this is where the assistance of taxonomists becomes very important. Next. In the past, taxonomists had to rely on light microscopy and stereo microscopes to pick out tiny details on insects using what at the time was microscopy, which was limited by having very low depths of field and not very high magnifications. And in working with tiny parasitic insects, such as we typically rely on in biological control, the old technology made this a very difficult task. Nowadays, taxonomists have additional tools with scanning electron microscopy and stacked image photography, which allows us to obtain very, very high magnifications, allowing the scientists to spot tiny differences that could be significant in helping to distinguish among these different species. Next. And the kinds of features that taxonomists are relying on are often very inconspicuous to the average person or the average scientist. Some of the features that are important in these groups of natural enemies include the position of hairs, the appearance of different parts of the body, the shape of these plates, and so on. Next. This slide shows two of the Leptopolina species that Kent mentioned that have been found attacking spotted wing in Asia. And again, these two parasites appear very, very similar to us. But it's crucial to be able to distinguish these, because species that appear very, very similar, even upon close morphological comparison, may have very, very different biologies and host preferences. And these differences in their biology can be crucial in being able to be successful in a biological control program, because these differences may influence their host preference, their habitat preference, or their climatic tolerance, and so on. Next. These are, again, this is a blow up of the two species just shown in the last slide. Next. And these circles just indicate the kinds of characters that taxonomists have shown to distinguish among these characters. And obviously this requires very, very high magnification and meticulous study of large series of specimens to be able to pick out characters that are potentially useful at discriminating one group or one species from another. Next. Fortunately, taxonomists now have another tool in DNA, which can be very, very useful in helping us to identify natural enemies. And modern molecular biology technology has advanced to the point where it's very, very easy to extract DNA from even very minute individual parasite specimens and quickly process this DNA and get an identification. A lot of this is now automated, so the process can be very fast. But by itself, even DNA does not necessarily tell us which species is different from another species or what the significance is. Next. So it's very important to integrate the results of genetic study, morphological taxonomy, with the results of experimental biology and observations from field biology. And together, this gives us a much better picture of what actually constitutes one species versus another species. Taking an example of this species that we're particularly interested in from Asia, Ganaspis brasiliensis, we now know, as the result of these cumulative different types of studies altogether, that within what appears to be one species, G. brasiliensis, there actually are 5 distinct genetic lineages. And 4 of these lineages do not attack spotted wing Drosophila, but they concentrate on attacking other Drosophila species. The one lineage that does attack spotted wing Drosophila does appear to be a specialist on spotted wing, and it attacks spotted wing and fresh fruit, which is very important for it to be considered as a useful biocontrol agent. Some of these other lineages are poorly adapted to D. Suzukii, and they're encapsulated, rendering them rather unimportant. Next. So as a result of these taxonomic studies, again, we now know that there are more than a dozen species that have been shown to be natural enemies of spotted wing in Asia. And because of our interaction with these different taxonomists, we know which of these species look like they are going to be the most effective, and that we're concentrating further attention on. And now I'm going to turn it back over to Kent to continue with the next steps.
Great, thank you, Kim. So now we're getting to some of the fun part. This is the quarantine work. We think we've done a fairly good job of surveying different regions in China. We still want to go to northern China, where they've got spotted wing, but levels of parasitism are different. We want to do climate matching to find out regions where they might match best to North America. For example, what might work in California may not work in Michigan. But the first thing we have to do is to show that these parasitoids are effective and that they won't cause any harm. Right now, they're in quarantine in Berkeley. Being in quarantine implies that they're not released into North America yet. It's an area that's got 3 doors to it before you can get inside. It's a USDA federally monitored facility. And so what goes in quarantine has to have permission to get out of quarantine. What I'd like to do is describe the studies that we've gone through to get material out of quarantine. And this is a close-up picture of Ganaspis brasiliensis. This is the lineage that Kim just talked about that is specialized on spotted wing Drosophila. This is a picture of it attacking spotted wing larvae inside a blueberry. You take these things and you put them in artificial diet with spotted wing, and they're not as effective. You put spotted wing into a blueberry or strawberry, these parasites go crazy. They really search that strawberry carefully, draining their ovipositor on. And what you see here in this picture is that insect is got that needle-like ovipositor right there. Perfect. Thank you, Hannah. Probing and getting down to that larva inside the fruit. So we're very excited about this one. The other parasitoid is Leptopolyma. This is also on blueberries, also attacking spotted wing. We're not quite as excited about this one because, as I'll show you, it may not be as much of a specialist, so it might be a little bit harder to get out of quarantine. So what are we doing in quarantine? So these are the kinds of studies. You can see Shin King, John, and Ali there in quarantine. We've got a section that's got glass walls on it. Of course, they're double paned. Again, it has to be at USDA certification, but we have to have the natural light come through because we're growing plants inside this to grow pests on to test these different natural enemies. These are our 3 larval parasitoids, Asobara, Leptopolyma, and Ganaspis. We also got these pupal parasitoids out, Pachycropoides and Trichopria, but again, because they're already in North America, we decided not to test those to see about releasing those in North America. So we're focusing on these larval parasitoids to add to what we don't have now. Our first tests have to do with biology and efficiency. We're doing studies like reproductive potential, olfactory response. How well do they search? Do they search over the fruit and get 100% of the spotted wing, or do they just attack one and move on? What host stage do they attack? Probably even more important is safety. We call this host specificity. Do they attack spotted wing Drosophila and everything else, or only spotted wing? So I can guarantee you, if they attack any native fly, if they attack anything that's considered to be beneficial, we'll never get permission to get this out of quarantine. We're also looking at climate tolerance or climate adaptability. So we put these parasitoids into temperature cabinets. We want to find out what range of temperatures they might be able to tolerate. Heather talked about Pachycropoides and the Trichopria, the pupal parasitoids. They're not found in identical areas in North America because they've got different temperature tolerances. So maybe Leptopolyma might work better in Michigan and Ganaspis better in California. That's what we're trying to find out. And then finally, potential interactions amongst different parasitoids. We don't want to release all 3 if they compete with each other when Ganaspis might be the best. We don't want to release anything that might negatively impact the pupal parasitoids we already have here. So all of these things are studied in quarantine to make a guess at what's going to happen if these ever get out of quarantine. So these are just 3 quick examples of the kinds of studies we're doing and some of the data we're collecting. All of this can be found in publications that are on the website. that Hannah showed you in the very beginning of this presentation. So for example, what host stage or host age do they attack? Assobara, a little bit more of a generalist. We've got 1 day old, 2, 3, and 4 day old spotted wing. It attacks them all, very aggressive. Again, in China and South Korea, we're finding this coming out of a lot of different plant material we're collecting. Ganaspis and Leptopolyma, we think these are specialists. These are really attacking the younger spotted wing Drosophila, the first day, second day old. They do attack the later ones, but not as preferred. Another example is lifetime fecundity. Assobara, the braconid, you see it comes out as an adult from its pupa, and it's got a lot of eggs inside it, the adult female, and starts depositing a lot of eggs right off the bat. As that female gets a little bit older, the egg deposition goes down lower and lower, so it's still alive, but probably only parasitizing 1 or 2 spotted wing per day. Compare that to Leptopolyma, the fecundity. It comes out and it's depositing eggs in the spotted wing, about 6 eggs per day throughout its lifetime. Overall, Assobara produced about 117 offspring per female and lived for about 16 days. The Leptopolyma and the Ganaspis, about 107 offspring per female, lived for about 20 days. This is all very good. Imagine if we can get one parasite killing about 120 spotted wing and producing about 120 offspring that go off then and kill another 120 each. So this is all good stuff. Functional response. The more spotted wing you've got, the more they kill. So we just give them a small number of spotted wing, they kill a small number, you keep on giving them more and more, they kill more and more. Both the braconid did this and the fecundity did this. This is another good thing. This just means that they're going to respond to spotted wing density in the field. If you've got a lot of spotted wing, they'll stay there and kill more. So these are all good host efficiency things. What about host finding? This is a Y-tube olfactometer. Basically, you put a volatile at one end and you release the parasite at the other end. It gets to that Y-tube and it tries to decide which way should I go. So when we look at these different parasitoids, and I'll just give you one example here. This is with Assobara. Next. We see here we've got blackberry, strawberry, and cherry. We've got healthy fruit, and we've got fruit infested with spotted wing Drosophila. We put the parasitoid Assobara at one end, and it makes a decision, and it almost always goes about 65 to 85% of the time, goes to the fruit infested with spotted wing Drosophila. We can use this data to make the argument that these parasitoids will find the spotted wing rather than going off and attacking something else. So all this is adding up to good information to get these out of quarantine. But the most important thing is this host specificity test. This is the safety test I mentioned. We see here just a list of different Drosophilids and closely related species in what's called a phylogeny tree, how closely related these things are. Circled in red is spotted wing Drosophila with its generic name, Drosophila suzukii. So what we want to do in a perfect world is these parasites would only attack spotted wing and nothing else. We tested these 24 different species. Next. And this is what we found for our 3 larval parasitoids. So we've got these different fruit flies or vinegar flies over on the left. Circled in red are this closely related group, spotted wing, melanogaster, and simulans. These are all very closely related. We look at Assobara, that's our braconid parasitoid, and we see it's attacking a lot of different fly species. To me, that means we're probably never going to get permission to get this out of quarantine. We look at Leptopolyma, it's really focusing on the suzukii group. It does attack some others, not as efficiently, but it does attack some other fly species. That's where information like the olfactometer comes in, because maybe they'll never find these in the field because they're geared up towards fruit. We look at Ganaspis, that's kind of our star. It really is only attacking the very closely related species. Down below, down here, we see it does attack one distant related species, but notice that only males, or mostly males, are really being produced from this. So even when it attacks something else, it doesn't really want to, and it puts a male egg in. So this is all adding up to good information. And I also want to point out these tests are being done inside these little diet dishes. So this is a physiological host range test, not an ecological host range test. So most likely in the field, these parasitoids would never find anything that, for example, is on the beach. Some of these flies might be coming out of something like seaweed. So we use all this information to put together a petition to request getting these things out of quarantine. So that brings me to my timeline. Where are we right now? We're actually making pretty good progress. Spotted wing was first found and identified in California in 2008. We initially started to look at insecticides, and that's common for all of these invasive species. That's typically what we do because we've got to develop IPM programs. In 2011, that's when people first started to look at natural enemies. Von Walton, Kim Homer, myself, Hannah, Barak, Yana Lee, Rufus Isaacs, Jeff, a number of people, Jeff Miller in Oregon, and Greg Loeb in Cornell. And so we were looking at North American parasitoids, but at the same time, the Oregon group started to also look for natural enemies in South Korea. So the initial collections were with Jeffrey Miller, Yan Soo from Korea, and Helmut Riedel. They were just trying to figure out where we could find, how we could collect. So they didn't bring back any parasitoids to quarantine. Next, 2014 was our third trip, and that's when I started to go on these things. And now we're starting to get some of these specialized fagitids, and we're getting enough to start doing our quarantine studies. Next. So 2015 is when I think we really started to get very serious about looking at this material in quarantine, and we focused on the Asobara, Ganaspis, and Leptopolyma. 2016, our collection in China, we hit the jackpot. We brought back over 1,000 parasitoids. We got great information from the country of origin that we can add to our petition. We got a wide range to give to taxonomists, as Kim Homer explained. And so next, we started the petition work. We submitted in July 2017 for Ganaspis and Leptopolyma. We got a review back about 6 months later. 5 of 8 reviewers approved. That's the good news. 3 of 8 reviewers denied the petition, but we believe we can answer all of their concerns. So we're working right now on climate matching, host specificity, and parasitoid taxonomy, and we hope to resubmit in July 2018 and get these parasitoids out of quarantine. So how will this work? Where will they work? An example from California, let's say you're a cherry grower. You're probably still going to spray your cherries. What's happening is spotted wing is coming from someplace else to your cherries, and then it's leaving your cherries after you harvest. Next, and it's going to other host plants. Spotted wing has this wide range. So where we're going to be releasing these parasitoids next will be in some of these non-crop areas. The idea is they're going to lower the pressure of spotted wing coming into your field. So let's say in a riparian zone, we can get 20%, 25% parasitism over the course of the year. With each generation, that's going to really lower the number of spotted wing coming into the cherries, the blueberries, the strawberries, into your field. That's going to make your pesticides more effective because you've got a lower density of pest pressure, and that's going to reduce the speed that we get resistance to insecticides and other things. So, summary. Currently, we really don't have effective biocontrols in the USA and probably Europe. We surveyed South Korea and China and found novel natural enemies. Next. Our quarantine studies show that the fagitids, more so than the braconids, are probably specialists, and this is important to get permits. We've requested our first round of USDA permission. The NAPO group denied our initial request, and so we're going to resubmit in 2018. We hope to get material out in 2019 to start doing some studies in cages, and then we hope to do widespread releases in 2020. It is a slow process, but the goal is to release only beneficial insects, so the goal really is to do no harm. So that's it from my end, and I'm going to turn this back over to Hannah.
Right. Thank you to all of our panelists. We will have a few minutes after the formal presentation to address any additional questions that have not already been answered in the chat box. For those of you who have not been looking at the chat box, Lauren has been going through and answering questions as we go, so you might want to take a scroll through the chat box and see what those responses have been before you exit the webinar. I did want to highlight some of our online resources. There's a number of different web pages that have been developed dealing with spotted wing Drosophila management and biology. Our project webpage is swdmanagement.org. A recording of this webinar will be posted by the end of next week, so you can go back and view this webinar again. And then we also have social media tools that you can encounter, both blogs and online through Facebook. Again, I want to highlight that this is a nationally collaborative project sponsored by USDA NIFA through the Specialty Crops Research Initiative program. And we've gotten support from a number of other agencies along the way as well. Finally, we have a short 4-question poll that we'd like to quickly get your feedback on this webinar, as well as to get opinions about future webinars that you would like to hear from our project. We've just popped this poll up here. Please go ahead and fill it out, and then we will use this information to get feedback. There are 4 total questions, so please scroll down all the way to the bottom to answer our 4th question as well. Again, thank you to my fellow panelists, Kent, Kim, Heather, and Lauren, our moderator. I'm going to go ahead and leave the webinar open for just a few minutes past 1 o'clock here, so that we can answer any questions that you guys might have. I'm going to invite Lauren to come over and read out a few of the questions so that our panelists can answer.
One of the first questions, one of the questions that I cannot answer, and I'm hoping Kent or Kim could answer this, is about how many generations of the specialist parasitoids occur before the pressure results in a shift in SWD adaptation or an alternate prey item. I don't know if anybody's looked into this or if there's an estimate we can pull off of other areas. Do you have any input on that?
So the question is, how many generations before you get a shift to spotted wing of some of these parasitoids?
I think it's before the parasitoids shift spotted wing to adapting to a different prey or a different host item.
So I think that it's been found a lot of times the gentlest natural enemies, the ones that Heather described, they will shift onto an invasive species because they kind of find that niche. In terms of the specialized natural enemies we're working with that evolved with spotted wing, it's an arms race. So if the spotted wing were to evolve to have a little bit better encapsulation of Ganaspis, then hopefully the Ganaspis evolves to fight that off. I hope that makes sense. So the idea with a specialized natural enemy is that it's going to stay specialized on spotted wing.
Yeah, I think that's, just to reiterate what Ken said, I think that's really the value of this classical biological control technique that both Kim and Kent described, where you do explorations in the native range of this organism. You're taking advantage of millions of years, hopefully, of evolution to select the least risky tools to bring into the United States. All right, other questions, Lauren?
So there's actually been some questions that I think will be addressed. They're very focused on cultural management and on organic management. So that'll be addressed actually next week by a team that we collaborate a little bit with. There's an organic research and extension initiative webinar, and I don't remember the date off my head.
So we have a parallel organic project that a number of folks from this Specialty Crops Research Initiative group are also involved in. And that group will be having their project webinar next week. We will send that link out to the email list for this webinar. So we'll send that out to you guys via email.
And I just want to answer one question too about the parasitoids being exposed to different pesticide chemistries. I'm sure that any of the broad spectrum pesticides will be harmful to these rockstar parasitoids that we're talking about here, as the question was posed. And that is something to do in the future. Right now, we simply cannot do it because everything's in quarantine, so we can't give anyone these species to test.
All right, and so we are at 5 minutes after 1 o'clock here. In the interest of everyone's time, I'm going to go ahead and end the webinar. Thank you so much for attending, and we do really appreciate your feedback. You are also welcome to email me any additional questions or comments that you have at the address shown on the screen, and thank you again. Can you stop recording?